Alright, we're going to talk about gametogenesis, and more specifically, oogenesis as well as spermatogenesis. And first, a little bit of vocabulary review. This N here is referring to ploidy. Remember that human organisms are diploid organisms. That means remember we have 23 different chromosomes, and you have one copy from your mom, one copy from dad, so you get diploid. The C is going to refer to the number of chromatids you have. And I was always confused between what a chromatid and a chromosome. So here is a chromosome before replication. And then after it replicates, this is what it looks like. The chromosome after replication. And it is still one chromosome here. This is still one chromosome. But now, what we call these two separate ones are two chromatids. They're sister chromatids. They're joined by the centromere. But remember that it's still one chromosome and one chromosome afterwards. So now we're going to start with this Oogonium. This is diploid, as you can see. There's one from mom and there's one from dad. See, mom, dad. And how many chromatids are there? There's one and there's two. So it's 2N, 2C. This is going to undergo mitosis and duplicate its DNA. And this is all happening in utero as a fetus. This is also when the number of your eggs are proliferating and increasing. And this goes to the max of 7 million. And this is all before birth. So birth is right here. Okay. And it's actually after that it's going to slowly decline until it's about 2 million. You don't have to memorize that. And then this, now you get a primary oocyte. And what is it? Is this diploid or haploid? You got one copy from mom, one copy from dad. So that's diploid. How many chromatids are there? There's 1, 2, 3, 4. 4 chromatids. So 2N, 4C. This is going to start meiosis 1. So the primary oocyte starts meiosis 1. And it actually stops in prophase 1. And this is all happening again before birth. So we get mitosis and meiosis 1. I misspelled it, but meiosis 1. It's all before birth, but it's going to stop in prophase 1. This is key. You have to remember this. They're going to test you on it. So it's going to stay in prophase 1, prophase 1, prophase 1, prophase 1. It's all prophase 1. See, I guess from all the way from here to here. It's going to stay in this phase until you get to this thing called puberty. And what happens in puberty is you start ovulating. So the egg that gets ovulated will finish meiosis 1. And so meiosis 1 finishes, so you get a splitting. And note there's some uneven splitting of cytoplasm. So this one is a lot smaller, has a lot less cytoplasm. This is called a polar body. This one, this greedy one that gets all the cytoplasm, is the secondary oocyte. And note that it is now haploid. There's only one copy of this chromosome. We were using chromosome 22 as an example. There's now only one chromosome of chromosome 22. This is from the dad in this case. So it's haploid. And there are still 2 chromatids, so it's 2C. And now this secondary oocyte will start meiosis 2. So primary oocyte starts meiosis 1. Secondary oocyte undergoes meiosis 2. And then this will stop in metaphase 2. Okay. So it's going to go into meiosis 2. But it's going to stop in metaphase 2 and stay there in that state until Mr. Sperm comes along and fertilizes your secondary oocyte. And at that point, it will finish meiosis 2 and it will split again. And note there's again a uneven distribution of the cytoplasm, with another polar body being generated. And finally we have an ovum that is haploid, and it is only one chromatid, so it's 1N, 1C. Alright, so now let's talk about spermatogenesis. It's largely very similar. I'm going to point out a couple key differences. Key difference number 1 is that this begins in puberty. Remember that oogenesis was largely happened in utero, and then completes it after you ovulate and then need some fertilization to go all the way. This begins in puberty, and it pretty much happens constantly. And the whole process to go from a spermatogonium to a sperm takes 2 months, okay? Spermatogonium to sperm. And it does not arrest at any point. And note that although it takes 2 months, you're doing this process continuously. So every second I'm counting 1, 2. You got millions of sperms getting produced, and it's going to take 2 months for them to eventually mature the whole way. And this is all happening in these seminiferous tubules inside the testes. And if we take a little cross section here, this is what we're going to see. And remember, we have the Sertoli cells here. And then these are nourishing the sperm and helping them develop. And now you start out with spermatogonium in the periphery. And as you go inside, they're going to mature until you become a sperm in the middle of the tubule. And now it's in the middle of the tubule. You can travel down that tubule as we just drew out, go to the epididymis, and it's still inside the tubule now. It's going to leave through the vas deferens, and that's where your sperm comes out, through your penis. So let's take a closer look. This is the tubule. I apologize for my poor drawing. And this is our Sertoli cell. And now we're going to look at the spermatogonium. And in this case, we're going to look at the X and Y chromosomes. I just wanted to illustrate it here. So remember, that's the 23rd chromosome. You have X and Y for males. This, again, is diploid because you have one from mom, you have one from dad. And what is the C? It's 1, 2, so 2N, 2C. It is going to undergo mitosis, and while it's doing that, it's going to cross something called the blood testes barrier. And this is formed by the two Sertoli cells, as you can see here. Sertoli cell 1, Sertoli cell 2, and this is the barrier. And this is to prevent crap in the blood entering and messing up your sperm production. You wouldn't want that. You want your sperm production to be nice and free of any other problems. So it's going to pass through the blood testes barrier, and it's going to undergo mitosis, similar to how the oogonium does it. And now you get a primary spermatocyte. So another test is this diploid or haploid. You see it's one from mom, one from dad. So you have diploid, and how many chromatids? 1, 2, 3, 4. So 2N, 4C. And this primary spermatocyte undergoes meiosis 1, and it's going to become the secondary spermatocyte. And note that as it does so, there's a nice even distribution of cytoplasm. These are equal in size. And then you very nicely undergo meiosis 2, and you finally end up with a spermatid. Again, there's no pausing, there's no waiting like there is in oogenesis. And this spermatid will finally mature through spermiogenesis. And spermiogenesis has 2 key parts. 1, you lose the cytoplasm. And 2, you gain the acrosomal cap. And if you remember, do you remember what the cap does? It's this thing right here, and it contains all those enzymes for the sperm to penetrate that ovum and cause fertilization. So that is it for gametogenesis.